let us continue the discussion on the cell signaling. So, after addressing the 2 points that I have mentioned in the last module, that what is the critical distance and how this signaling molecule concentration at the target cell both are important in determining the cell communication. Now, the next step, once these 2 questions are addressed, the next step of understanding would be how cell surface receptors would be linked or would be tagged or would be hooked to signaling molecules. Now, there are 2 type of signaling molecules that has been shown in this particular slide. One is the the hydrophilic signaling molecule, and another one is that small hydrophobic signal molecule. If there is small hydrophobic signal molecule, first of all because of their hydrophobic nature, and secondly because of their small sizes, they can be internalized directly into the target cell. So, this is your target cell here. So, this is your target cell T. So, this is your signaling molecule that we are talking about, this is small hydrophobic. So, because of these 2 reasons, they can be transported, they can be internalized into the target cell rather very easily. And again through the nuclear pore complex, if you remember here in the nuclear membrane, you have a very characteristic nuclear pore complex. Through this nuclear pore complex, the signaling molecule can be internalized well within the nucleus also, and therefore, after it can cause some changes in the gene expression of the target cell. But the story is little different when we consider that signaling molecule is hydrophilic in nature and it is little bit larger in size compared to the small hydrophobic signal molecules. Then the only option that is biologically possible, that is it will be hooked to the cell surface receptors, and therefore, this binding efficiency comes into picture. So, this is your binding here. So, binding essentially is established between the hydrophilic signal molecule and the cell surface receptors. So, certain comments that is mentioned here, that most signaling molecules are hydrophilic in nature and therefore, they cannot cross physically to the plasma membrane directly, and in turn they are bound to the cell surface receptors, which in turn generate one or more signals inside target cell. The third point that has been described with reference to this particular scenario, that when there is a small hydrophobic signal molecule, then it can cross the plasma membrane, both of the cell membrane and also in the nuclear membrane, and can directly be even internalized into the nucleus itself. So, that once it is once this hydrophilic signaling molecule is attached to the cell surface receptors here. So, that is the cell surface receptor, that is a kind of transmembrane protein. Then what will happen? This attachment will further sense out the, or which we will further activate the downstream intracellular signaling proteins. So, these intracellular signaling proteins are shown here by different geometric shaped objects like rectangle, triangle or circle. So, essentially these different shape indicates that these proteins, that intracellular signaling proteins will have either different chemistry or different conformations, that is within the cytoplasm of the cell. So, if I may show you the target cell. So, this is your target cell membrane and this is your cytoplasm of the target cell. So, these entire things what I have shown here, this entire thing is happening well within the cytoplasm of the target cell, and this is your outside the cell, that is the extracellular space. So, some of the comments that I may like to make here, that the receptor ligand complex, once it is internalized, So, signaling molecules then dissociates from receptor and receptor recycle to cell membrane. A typical time constant for internalization of the signaling molecules is of the order of 15 to 30 minutes. What it means is that, so once this biological process gets activated, you have to know that what is the time scale over which these intracellular signaling proteins are activated, and this time constant is somewhere between 15 to 30 minutes. So, within half an hour time period, the signaling intracellular signaling proteins or downstream signaling proteins are activated within the target cell now. So, this is your target cell. Now, if the signal transduction process has to activate or has to cause certain gene expression changes, that means this has to enter the nucleus of the cell. So, suppose this is your nucleus and this is your nuclear membrane, and there you have a nuclear pore complex is nuclear pore complex is present. So, that means this one of the signaling proteins have to now travel through the nuclear pore complex to well within the nucleus, so that these gene regulatory proteins are now activated, and the end result or the end point objective or end point result is that this leads to altering of the gene expression. So, now from the basic description of this process you can see, now if gene expression has to take place, then 15 to 30 minutes time scale is too small. So, this entire process up to the intracellular signaling proteins to activated and up to the gene expression will take few hours to few days. So, therefore, normally cell differentiation assays or cell differentiation process takes place from few days to few weeks timeline, whereas simple metabolic activity or cytoskeletal reorganization can take place from minutes to hours or maximum to a few days in time. So, certainly if any alteration of certain metabolic activity or to the cytoskeletal reorganization has to take place within the target cell, that takes place in a much shorter time frame than gene expression if the cell signaling has to activate or has to lead to the gene expression changes. So, this is mentioned here in this particular slide. Other things that has been mentioned here, 10,000 to 70,000 growth factor molecules need to be internalized to stimulate the cell division process. So, that means somewhere around 10 to the power 4 number of molecules are to be internalized, so that cell division processes has to be activated in the target cell. Okay. What are the more specific details of the cell signaling proteins that has been summarized in this particular slide? So, what you see here, it is a very complex signaling processes that has been shown here, and in fact it is a very busy slide. So, let me spend some time to explain to you in a little bit briefly here. So, what you see here, that you have a extracellular signaling molecule and you have a cell surface receptor proteins in a target cell. Okay. This is your target cell. Now, once this, so that moment that extracellular signal molecule get attached to the receptor protein, the switch is on. It is like you put your switch on in your home, so that light is on. So, similar analogy you can bring in here to understand this cell signaling process in a much more better manner. So, I repeat that the moment the extracellular signal molecule which is being transported from some far away from the source cell to the target cell here. Now, if the moment that extracellular signaling molecule gets hooked to the transmembrane or receptor protein of a target cell, this target protein will be now switched on, and not only this target protein will be activated, but this entire signaling pathways would be slowly switched on or would slowly be activated. And as a result, the signal transduction processes will start taking place within the target cell, and as part of the signal transduction processes, a number of downstream signaling proteins like relay protein, like adapter proteins, which are shown with different geometric object here, that would be now activated. So, the arrow essentially indicates, this kind of downstream arrows essentially indicate that sequentially one by one these signaling proteins are now slowly gets activated. This slide also mentions something called scaffold protein. So, scaffold protein essentially houses more than 2-3 different types of proteins, which are essentially relay protein or adapter protein, and this relay protein adapter proteins take active part in the intracellular internalization of the signaling processes, which essentially convey certain information or certain communication communication from the source cell to the target cell. Now, there is also certain other proteins which are present in the cell within the cytoplasm also. It is like bifurcation protein. Bifurcation protein means the protein which will convey or which will channelize in two different ways from the adapter protein itself. And the other types of proteins which take active role in the cell signaling processes are called amplifier or transducer protein. The name amplifier suggests definitely certain signaling molecule concentration will now be amplified. What it means is that this large signaling molecule will activate very small intracellular mediator proteins, and you can see from the basic physical size of these small intracellular mediator proteins that a large number of intracellular mediator proteins can be integrated to activate what is known as the integrator protein. So, the role of integrator protein is to integrate all the information or all the signal intensity in the form of a anchoring protein now, and this anchoring protein will be hooked to a modulator protein, and if the gene expression is to be altered, then finally the messenger protein will activate certain target protein well within the nucleus, and this target protein essentially will activate what is known as the transcription process in the nucleus itself. So, the transcription process essentially defined is that transformation from DNA to RNA within the nucleus itself, and ultimately that once this RNA forms, again RNA is getting out of the cells through the nuclear pore complex, and outside the cell nucleus in a eukaryotic cell, RNA can again transforms to protein. So, what is known as the translation process. So, this entire transcription and translation process, and many times reverse transcription process, like it goes from RNA to DNA, all these processes are being activated once the appropriate signal transduction mechanism is activated well within the target cell. So, from this previous description, it should be clear to you that speed of a response to an extracellular signal depends not only on the signal delivery, mechanism, but also on the nature of target cell response. So, that means that if a target cell responds to the external signal much faster, then all these processes what I have mentioned like 15 to 30 minutes can be even little shorter also. So, the two things that is important, that how target cell is receptive or is sensitive to a particular signaling molecule that it receives from a source cell, and therefore, what kind of downstream signaling proteins are activated and to what kind of complex communication network is also being activated downstream in the cytoplasm. So, that ultimately results in what is the time scale or what is the response time scale of the target cell itself. If a target cell response requires only changes in the already existing proteins in the target cell, then it can occur seconds or milliseconds, and when the response involves gene delivery, gene expression or synthesis of new proteins, then it usually occurs hours irrespective of the signal delivery mode. These are the different proteins I have mentioned in the last slide, and in each proteins actually they have some specific role to play. When there are multiple amplification steps in a relay chain, the chain is referred to as signaling cascade. So, signaling cascade means when there are multiple proteins are involved, then it is called a signaling cascade, which is activated in the target cell itself. Okay. So, detection, transmission. So, detection means what is the so detection means that what is the specificity or what is the sensitivity of a target cell to a specific signaling molecule. So, what is the specificity of this target cell, that it will sense this specific signal molecule which is which has been diffused from the source cell at the target cell surface. The second thing is the transmission. Now, how the signal transmission takes place, within the target cell once the cell surface receptor protein is activated or is switched on. Third thing is the amplification of signals. Amplification means, if you go back to this particular slide, you can see that there is certain amplifier protein or transducer protein is being activated, and this will activate in a turn the small intracellular mediator proteins. So, this signal transduction network therefore can be represented as a components of a complex interconnected circuit. And what is the elements of the circuit? Like if you are electrical engineering, you know that there is resistance, capacitance, inductance, these are like different components of electrical circuits. So, therefore, equivalent to the electrical circuit description, you can also see that whether this relay protein, adapter protein or amplifier protein, what kind of role it plays, whether it will resist the way the signal transduction process takes place, or whether it activates, or whether it is more act like a capacitor or amplifier. So, all these kind of things that can be described by a complex interconnected circuit. The other things that is important that I will explain to you next slide is that cellular behavior like cell survival, proliferation generally stimulated by a specific combination of extracellular signals rather than by a single signal. Let me substantiate this statement in the next slide. What it means is that, suppose this is one specific cell, and this specific cell is receiving 3 type of signaling molecules from its cellular microenvironment, that is the A, B, C. These are 3 different type of signaling molecules, and under the receipt of these 3 different signal molecules, this cell now survives. So, essentially cell survival depend on not only one type of signal molecule, that is A, but on 3 different type of signal molecules, that is A, B, C. Now, once the cell survives, then cell has a tendency to divide into 2 daughter cells by a process called cytokinesis mitosis, and this is your mother cell and this is your 2 daughter cells, and this mother cells to 2 daughter cells, that means cell division process has to take place, and cell division means you need some extra signaling molecules here, that is D and E. So, the rationale behind this one is that, first of all, cell has to survive, and therefore, you need A, B, C 3 different type of signal molecules. Now, once the cell has to divide to 2 daughter cells, that means cell needs some additional signaling molecules, so that it receives the instruction for its division to 2 daughter cells. So, therefore, cell division, the signaling molecules or signaling network of the cell, for the cell to divide, essentially needs the molecules which are essentially required for survival plus the molecules which are required for cell division. Now, third thing that I have mentioned here is the differentiation. So, differentiation means it not only needs to survive, but also it needs to change its gene expression profile. Therefore, it goes to a more matured mature cell type, which will have a altered, which will have a different cellular functionality. So, therefore, differentiation needs two again additional two signal types, that is F and G, which is different from that of the signaling molecules which is required for a cell to divide. So, A, B, C, that is 3 for the cell survival, and F, G, these are like specific signal molecules which will stimulate the cell to differentiate to a matured cell type. So, now you have a 5 different type of signaling molecules, one for survival and one for differentiation. Now, under certain circumstances in any cellular microenvironment, if you kind of remove all these 3 signaling molecules A, B, C, then what will happen? Now, cell is not receiving any signal molecule, and therefore, it will activate its own its own suicidal mechanism, and then which is known as an apoptosis or programmed cell death, and that will be reflected in a very characteristic morphological changes. As you can see here, it will be shrinking, blebbing, and this apoptotic cell. So, the take home message from this slide essentially is that all the cell behavior is stimulated by a specific combination of extracellular signals rather than a single cell, and a cell therefore has to integrate all these signaling molecules and then integrate, and therefore and thereafter it can survive. Now, for a cell to divide, cell has to integrate A, B, C, D, all the 5 type of signaling molecules, and then final endpoint result is the 2 daughter cells. Okay. So, the integration usually depends on the integrator proteins, and these integrator proteins I have shown you before. These integrator proteins are also present in the cytoplasm. So, from different places the cell is receiving A to E, let us say 5 different proteins here, signaling proteins. So, essentially that all these different proteins once it receives, so all 5 different signaling pathways now activated. Now, A, B, C, D, E, all 5 signaling pathways now finally should be linked to a integrator protein, which will integrate all the information and then finally will give the cell the required instructions for its division process to activate. And they require multiple signal inputs to produce an output, and that causes the desired biological effect. So, what I mentioned here, so suppose you have a eukaryotic cell and this is your target cell. Now, target cell is receiving different signaling molecule of different type, let us say A, B, then C, then D, and then E, all different signaling molecules. So, all these signaling molecules will now get attached to a different type of transmembrane protein, and then for simplicity or just to show the distinction between different signaling to show the different type of signaling molecule, I am showing you the different signaling molecule by different geometric object, and so one is a circle, one is the rectangle and all. Now, in the target cell, now each time, so all the 5 different signaling molecules, once they are hooked to cell surface receptors, then they will essentially activate, or or 5 different signaling pathways will now be switched on. So, all these 5 different signaling pathways is to be now integrated by a protein called integrator protein, and this integrator protein is also present within the cytoplasm of a target cell. So, the role of integrator protein is to integrate all the signaling information into a single information, and that single channel now will be activated and the net result can be either cell differentiation or can be cell division. So, this is what has been, this is what can be summarized from the cell signaling and cell signal transduction process. Some examples of the influence of the cell signaling. So, you have a fibroblast cells, fibroblast is the connective tissue cells, and you have a nucleus, and then it can either go transdifferentiation to smooth muscle cells. Now, smooth muscle cells are present in the typical muscle tissue as well as the cardiac tissue also, or fibroblast can go can undergo transdifferentiation to osteoblast, that is the bone forming cells. I think I have mentioned in some of the earlier modules that osteo means bone, osteoblast means bone formation cells, osteocytes means that is the matured osteoblast. So, when a stem cell will undergo osteogenesis or will undergo differentiation through osteoblast lineage, then the end member of the osteogenesis is the osteocytes, which are the more matured osteoblast cells. The stem cells also the fibroblast also can undergo transdifferentiation to cartilage cells, that is the chondrocytes, or fat cell, that is the adipocytes. So, this is the interconversions that are thought to occur that can take place within the family. So, these key points from this so much discussion that I had in these last two modules can be summarized here, that cellular response like proliferation, growth can be stimulated by specific combinations of extracellular signals rather than a single signal acting alone. A cell may be programmed to respond to one combination of signals by differentiating, or to another combination of multiplying, or to another performing to specialized functions such as contraction or secretion. Let me go back to these schematics that I have drawn for your understanding a few slides back. The schematic essentially shows that a target cell is receiving 5 different type of signaling molecules A to E. Now, this A to C is required maybe only for survival itself. Now, this DE can be two different kind of cell, 2 different kind of signaling molecules. So, the DE can be different type of signaling proteins, and the DE, the integrator proteins within content within the target cell will now integrate all the informations or all the different signaling pathways or downstream signaling proteins which are activated as a result of receiving 5 different signal molecules, and therefore, will instruct the cells to either divide or differentiate. But my point was that for division, this DE can be 2 different signaling molecules. For differentiation, that these DE can be 2 different signal molecules. So, this point needs to be borne in mind that different type of signal molecules are needed for cells to perform different type of functionality as a result of the cell signaling process. Okay. Most of the cells in a complex signaling environment are programmed to depend on a specific combination of signals simply to survive, and when deprived of the survival related signals, a cell activates suicidal program and kills itself, a process called as a programmed cell death or apoptosis. So, this is this has been mentioned also here in this particular slide. So, I reiterate my earlier statement that a given cell has to receive the 3 different signals A, B, C. Now, under certain circumstances in a cellular microenvironment, if a cell does not receive any of the signaling molecule either A or B or C, then what will happen? Cell would activate its own suicidal mechanism, and therefore, cell will undergo programmed cell death, which is known as apoptosis in cell biology. So, I think I stop here, and the next module I think I will start with the cell fate processes.